Assalamualaikum, my name is Tizukrina. So today I will present about the detection of viral disease in tilapia farm. So for the introduction, a sudden death of tilapia in a tilapia farm can cause a major loss for the farmer and affect our country's aquaculture. If the sudden death of the tilapia is in a large amount, it can be caused by a lot of causes, but the most threatening is tilapia lake virus disease, or as known as TILV. So TILV is a new disease which is first identified in 2014 in Israel. So this disease spreads really fast like wildfire, and it has spreaded across border, and this causes tilapia mortality up to 90%. Therefore, sudden death of tilapia with large amount must be examined for TILV to avoid this mortal disease from spreading into our ecosystem. So these are the issues in our aquaculture. So there are a lot of issues regarding sudden death of tilapia in large amounts, and this is the method that we are avoiding, which is this TILV spreading into our ecosystem, where tilapia were found dead in a recreational park. So my problem statement is sudden death of tilapia with large amount is sent for detection of TILV by RT-qPCR or RT-LAMP in order to take correct measurement to avoid TILV from transmitting into the ecosystem. So my objective is to detect the presence of TILV by RT-qPCR or RT-LAMP in Tilapia oricormis niloticus. These are the two articles that I have chosen, where article 1 is Tagman RT-qPCR assay for Tilapia lake virus, and the second one is rapid detection of tilapia lake virus using a one-step reverse transcription loop mediated isotomal amplification assay. So the methodology for the first article is, first of all, is sample collecting. Second is total RNA extraction. Third one is Tagman assay design and conditions. And the fourth one is repeatability, reproducibility, and specificity testing. So the second article is, the first method is experimental design and animal protocol, and the second is RT-LAMP primer design. Number 3 is RNA isolation and cDNA synthesis. Number 4 is RT-LAMP and LAMP reaction. Number 5 is analysis of the RT-LAMP and LAMP products using gel detection and calcium detection. Number 6 is reverse transcription quantitative polymerase chain reaction, PCR, qPCR, and number 7 is sensitivity and specificity of RT-LAMP and RT-qPCR. So these are the results obtained in article 1. There are application curves of Tagman, standard curve of RT-qPCR amplification, partial multi-sequence alignment of segment 3 of tilapia lake virus, repeatability and reproducibility of the Tagman based RT-qPCR, specificity of Tagman based RT-qPCR for TILV detection, and for the next result is comparison of the sensitivities and quantitative power of the Tagman and SYVR green RT-qPCR assay. And lastly, for article number 1 is detection of TILV infected fish collected from different geographic location using Tagman and RT-qPCR. And the result for article number 2 is specificity of RT-LAMP assay for TILV detection through agarose gel electrophoresis. And the second one is amplification of product of fish tissues infected with other bacteria or viruses, and comparison of sensitivity of RT-LAMP and RT-qPCR, and detection of TILV in fish and cell culture samples using LAM and RT-qPCR. So the discussion for Article 1 is TILV is an emerging highly contagious disease caused by a novel orthomyxo-like virus in tilapia. So in this study, they have delivered a sensitive, rapid and specific Taqman probe-based RT-qPCR assay for the detection of TILV in field samples. So this development of high-performance diagnostic tools is of utmost importance for the implementation of screening and containment measures. This novel Taqman RT-qPCR method can be used for diagnostic purposes. This novel Taqman RT-qPCR method can be used for diagnostic purposes or in basic research aiming to better understand TILV, as well as in epidemiological studies of TILVD. And for Article 2, practically, a desirable diagnostic test is an assay with good sensitivity, specificity, simplicity, and immediate result interpretation. This result developed an RT-LAM protocol for the detection of TILV in fish tissues and cell lines. Compared with RT-qPCR, the RT-LAM assay offered high sensitivity and reliability in the diagnosis of TILV infection. Moreover, the RT-LAM assay could be applied to detect the disease in resource-limited countries where there is an urgent need for rapid diagnostic assay to guide TILV control. And these are my references. That's all from me. Thank you.